Hello everybody, welcome back to our channel. Okay, biology again. Previous week we talked about transport number 1. Today we talk about transport number 2 for AHL only. Okay, so work hard. Okay, we can learn a lot about the transport. First is the heart, and then is the plant. For the heart, for the circulation of the blood. Okay, we talk about the release and reuptake of the tissue fluid. For some reason, you'll learn later. Okay, so about the tissue fluid, because you remember, capillary has a high pressure, right? So the tissue fluid, the plasma may be squeezed out from the capillary and become the tissue fluid, and the tissue fluid had to be reabsorbed. There's some way to reabsorb it. First of all, at the arteriole on the left hand side arteriole, okay, the pressure is high, the tissue fluid will be squeezed out. Okay, but at the right, at the ring nerve, and then the pressure is low, and then the tissue fluid will go back inside. Tissue fluid is similar to the red blood, for the blood. Okay, very similar. Okay, so it's one way. Okay, natural way. And the composition of plasma and tissue fluid. Okay. Both blood contain red cell, plasma, and the plasma. Okay. The composition of plasma and tissue fluid are nearly identical. Okay. They both have oxygen, carbon dioxide, water, glucose, salt, protein, hormones, and enzymes. They are nearly the same, just don't want it to be too much. Okay. So exchange between tissue fluid and cell, actually maybe a part of the process for the carbon dioxide and oxygen to transfer, okay, by the tissue fluid. Okay. The plasma passes through the thin wall of capillary and becomes a tissue fluid. So this tissue fluid that releases oxygen to the cell, and it is this tissue fluid that gets the carbon dioxide from the tissue. Okay. This is the way of transfer. Okay. Of course, sometimes it is not perfect. Maybe the tissue fluid is too much, maybe high pressure and low pressure. Maybe high pressure squeeze out too much blood. Okay, the plasma. Okay, so they need another system to get back the tissue fluid. We call it lymph system or lymphatic system. The lymph system is another thing inside our body, and it drains the leak tissue fluid back to the blood circulation. Okay, so the excess tissue fluid is drained into the lymph ducts. Okay, and the lymph ducts has a thin walls to allow the drainage of tissue fluid to it. Okay, and lymph ducts also have valves because it's low pressure. Okay, to prevent the backflow of lymph. So remember, the tissue fluid can go back to the ring node. If can't, okay, then go through the lymphatic system. Okay. And when we talk about single and double circulation of the heart, okay, first of all, we learn something new, which is the fridge. Okay. For the fridge, and inside is another thing too. For the fish, it has only one single circulation for the bony fish. Okay, so the heart, from the heart, pump the blood to the gill to get the oxygen, and then go through the whole body and then go back. Okay. Bony fish has a single circulation system. Okay. Its heart only has two chambers. Okay. Its heart pumps the blood to the gill, then to the body, and then back to the heart. Okay. Artery carries blood out of the heart. Vein carries blood back to the heart. The same, but just one set of artery and vein. Look for the inside, even more funny. Okay, inside has an open circulation of blood. Okay. But also circulate back to the tubular heart. Okay, its heart does not have separate chamber. Just its heart just pump out the blood, and the blood will circulate around the whole body and then go back. And for the double circulation of mammal, why mammal need the double circulation thing? Okay, because mammal has two tasks. Okay, first, mammal's lung. Okay, cannot withstand so much high pressure. Okay. So lung has to be a little bit low pressure. But for the whole body circulation, the blood pressure has to be high. So these two contradictory requirement, making four chambers and two double circulation of blood. Oxygenated blood flows from the lung through the pulmonary vein to the heart, and then through the aorta to the body. Right? The oxygenated blood flows from the body through the vena cava to the heart, and then through the pulmonary artery to the lungs. Okay. And blood in the pulmonary ring is fresh from the lung. It has the highest oxygen concentration and lowest carbon dioxide concentration. Blood in pulmonary artery is heading towards the lung. Right? So it has the lowest oxygen concentration and the highest carbon dioxide concentration. That's a reverse. Just remember the throat. Just remember where is the lung. Okay. To the lung, lowest oxygen, highest carbon dioxide. From the lung, highest oxygen, lowest carbon dioxide. Okay. Essential of double circulation, yes, we have explained it. The lung because a low pressure of blood. Okay. If only one circulation, the high blood pressure will hurt the lung. Okay. not the cure. Okay. And then he need a high pressure to pump blood through the whole body. Okay, so low blood pressure blood from the heart, from the lung, cannot continue to the tissue of the whole body. Okay, the oxygenated and the deoxygenated blood are separate. Okay? Tissues of the whole body receive blood with high oxygen content. Okay. And then William Harvey is a Greek scientist due to also the discovery on heart and the blood flow. Okay. In ancient Greek, okay, Galen proposed blood simply flowing through the pores between the right and the left atria of the heart, okay, not pumped by the heart. Okay. The heart has a function of pumping blood. But William Harvey did not lots of work on circulation, lots of in the 17th century, 180 something, and then he found out blood is circulating and it's not consumed. Harvey estimated the amount of blood pumped out from the heart in half an hour. He found that it was larger than the body, whole body. Oh, crazy! So he concluded the blood must come back. Okay, so it's circulating, not consumed. And then he found out heart pumps of blood. Okay. In anatomy animal experiment, he found that the animal's heart swelled, becomes big. Okay, when the artery were tied. He concluded the blood is pumped out from the heart through the artery. And then unidirectional flow of blood is a human experiment, simple one. From our point of view, but it is innovative for William. Okay, and unidirectional flow of blood. Okay, human arm experiment. One side is tied. Okay, when the valves were shrunk. Okay, he found that he can't push blood down the forearm and on the wing, but he can push the blood up the forearm easily. So he concluded that valves ensuring that unidirectional flow of blood. And the blood vessel with valves flow blood back to the heart. Okay, so we find out the valve of the concept because one way is okay, the other way can't. Okay, so blood is blocked, back forward blood is blocked by the valves. Okay, and adaptation of the heart. Okay, so first of all you need to load the heart, left and the right. Okay, and first of all you need to load this is anatomy graph. Okay, and you imagine a human lying down and being dissected. So the chamber on the left of the graph actually is the right of the body. Okay, so left and right sometimes is hard to remember. Just name it according to the real one. Okay, and 4 chambers are there. Okay, so left atrium, left ventricle, right atrium, and right ventricle. Okay, 2 atria and 2 ventricle. The atria is larger, ventricle is smaller. And then look it up with the vena cava and inferior vena cava are connected to the right atrium as shown from the body. Okay, and pulmonary artery connected to the right ventricle as shown to heart to lung. Okay, pulmonary ring connected to the left atrium as shown from the lung. Okay, aorta is the largest artery because it's pumped the blood to the whole body. Okay, from the left ventricle. Aorta is it. And atrioventricle valves is the valve between atria and the ventricle is here. There are 2 kinds, okay? Bicuspid and tricuspid, okay? Bicuspid and the tricuspid one, okay? You need to remember the left one on the right is the bicuspid, okay? And the right ventricle one is the tricuspid. Okay? You have to distinguish them. But for the semilunar valve, they're the same, okay? So in IBDP examination, the substance carried in the atria and the ring are oxygen, carbon dioxide, antibiotics, nutrient, glucose, protein, hormones, and water. But the blood itself is the carrier. They are not being carried. For the plasma, plasmat, and then red blood cell are parts of the carrier, but they are not being carried. It's a distinguished definition in IBDP. Okay? And maintaining a high blood pressure, how to do it? Okay? High blood pressure is created by the contraction of the ventricle. Okay? The heart pump out the high pressure blood through the arteries. Okay. The muscular wall of artery is thick to resist the high pressure and prevent leaks. Okay. And the elastic vacuole and then the fiber on the arterial wall help pushing the blood. Okay. The lumen of the artery is narrow to maintain the high pressure. On the contrary, the wing has a wider lumen to decrease the pressure. Okay. We done it before, but now we want to stress on it. Okay. Stage in the cardiac cycle. You need to remember the sequence. Okay. Role of the heart in the blood from the lung to the left atrium, bicuspid, left ventricle, semilunar valve, body, right atrium, tricuspid, and then semilunar valve, right ventricle, and then lung. The sequence you need to remember clearly, okay? And remember left ventricle, okay, pumps blood to the whole body as the thickest wall, okay? Oxygenated blood flows through the left chamber. The oxygenated blood flows through the right chambers, okay? The two atria collect blood from the two wings, okay? Impulses are sent from sinus atrial nodes, okay, to muscle to initiate contraction. Contraction of skeletal muscle pushes the blood out from the heart. Okay. So we have a three phase. Okay. Phase one, okay. Atria systole. Okay. The contraction of atria pushes blood to the ventricle. Okay. At the same time, the ventricle are relaxed. Okay. The atrial ventricle valves are open and the semilunar valves are closed for the ventricle to be filled with blood. Okay. Second phase, ventricular systole. Okay. The contraction of ventricle pushes blood out from the heart, okay, to the lung and to the body. At the same time, the atria are relaxed. The atrial ventricle valves are closed to prevent backflow of blood, and the semilunar valves are open, okay, for the ventricle to push the blood from the heart. Okay. That's true. The blood flows through the lung and the body, okay. So at the same time, the semilunar valves are closed to prevent backflow of blood, okay. Both the atria and ventricle are relaxed. So remember the sequence: contract, relax, 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 contract, relax, open, close, open, close, open, close. Impossible to remember, so you need to remember the story, the three stages, three phases. And then even later for the SL lecture, we need to remember this chart about the pressure. In phase one, okay, remember broken line is the left atrium, solid line is the left ventricle, dotted line is the aorta, okay. For the phase one, okay, left atrium contract and pushes blood to the left ventricle. So pressure in the left atrium and the left ventricle rises first, okay. But still the atrium has to be higher to push the blood downward. As the left ventricle fills up the flow of blood in the left atrium, so the pressure in the left atrium decreases with that in the left ventricle until they are the same. They're the same, no more flowing of blood through them, okay. And the aorta has a higher pressure because than the left atrium to keep the semilunar valve closed, okay, such that the ventricle can be filled. In phase two, a left ventricle contracts, okay. Its pressure rises quickly, okay, crazy, okay, to keep the semilunar valve and to push the blood through the aorta. So the pressure of the left ventricle and the aorta becomes similar, okay, so they overlap, okay. Semilunar valve is closed, okay. Pressure in the left atrium remains low, okay. In phase 3, a left ventricle will stop pushing blood out of the aorta, just let the blood flow to the heart and to the body, okay. And pressure in the aorta decreases, okay. Both left atrium and left ventricle relax, their pressure drops to low. And blood flows through the aorta, the pressure of aorta remains a little bit higher, okay. Hypoxia, okay. The right ventricle pumps blood to the heart to get oxygen. Under hypoxia, okay, the body does not have enough oxygen to grow. Okay. To adapt with hypoxia, the right ventricle grows bigger to pump more blood to the heart to get oxygen. How do you know? Okay, so we do some experiment with the rats, okay. Not with him at all, of course, okay. Synoatrial look, okay. This is a SA look, okay. SA look is specialized tissue on the right atrium, okay. And the synoatrial look acts as a pacemaker to control the regular heart rate, okay. Synoatrial look send out electrical signals stimulating contraction of muscle. The electrical signal first reaches the wall of the atria, so atria contract first, okay. And then the electrical signal arrives the wall of the ventricle, so the ventricle contract next. Okay. And after that, both of them are relaxed, okay. So atria, ventricle. And the medulla of the brain has two nerves connected to the synoatrial loop, okay. For these two nerves, okay, medulla send impulses to the synoatrial loop to increase or decrease the heart rate, okay. Very funny. One nerve increase the heart rate, and the other decrease it. Epinephrine, also called adrenaline, is a hormone secreted to the bloodstream. Okay. When you do a vigorous physical exercise, okay, medulla of the adrenal glands secrete epinephrine. Epinephrine increases the rate and force of contraction of the heart. Okay. More blood flows through the body to deliver more oxygen and get back more carbon dioxide from metabolism. Okay, blood pressure also increases. At the uptake of mineral ions, and then we talk about the roots. Okay, the plants, xylem, phloem, transpiration, and now we add translocation. At the uptake of mineral ions, we know it. Okay, yeah, generation of root pressure in xylem vessel by active transfer of mineral ions. Okay. So the mineral ions in the soil are leaching into the plants. Okay, for example, nitrate, phosphate, calcium, magnesium, iron, potassium, and so on. We have to get it. Okay, these minerals are dissolved in water. So how can it be dissolved? How can it be absorbed by the roots? By active transfer. We'll talk about the process in detail, but hydrogen gradient actually. Mineral enters the plants through active transfer of ATP, which has a rich hair with many mitochondria to provide energy and ATP for active transfer. So when the mineral ions are going inside the roots, okay, the mineral ions near the roots becomes lower concentration than far from the roots. So mineral ions will continue to flow to the roots by diffusion, okay, easily, okay. And the roots has a long branch to increase the surface area absorption of mineral ions. And then in details, now we need to learn how this active uptake happen, okay. Actually, it's the energy where is the energy used. The energy is used to pumping the hydrogen ion out from the roots through the hydrogen pump. Okay, so hydrogen pumped out from the roots. Okay, so outside the roots there is a higher proton concentration, higher positive ion concentration. Okay, so the positive mineral ions diffuses into the roots through this proton gradient charge difference. Okay, and negative mineral ions linking with the hydrogen ion to diffuse into the root cell is a co-transport. Maybe you can see in some way. Okay. Difficulty of forming a root pressure sometimes because the transpiration is too low. Transpiration is insufficient. Why? Too humid surrounding, and the leaf is not open yet. Okay, in the spring. Okay, the leaf in the plants haven't opened in the spring yet. Okay. And then we talk about translocation. Translocation is the transfer of sugars, okay, the food, okay, from the source to the sink, opposite to the transpiration. Translocation is the transfer of organic compound, okay, from sources to sink in a plant, okay. Organic compound from the sources, for example, sugars, okay, from the liver, pumped to the phloem by active transfer. Can you guess about how to do it? Hydrogen ion again, very similar to the roots. Okay, the phloem beside the source has a high concentration of organic compounds. Okay, so low concentration of water, so the water moves into the phloem from the source by osmosis. Okay, but water is incompressible. So when when more water moves inside the phloem, a higher hydrostatic pressure is developed. So this higher hydrostatic pressure pushes the organic compound down to the whole plant. Okay. So the hydrostatic pressure, the gradient between the source and the sink, pushes the water with the organic compound from the source towards the sink. Very smart design. Okay. So how does it happen? Okay. How does the sugars is being actively transported from the source to the phloem? It is by active transport. The process is like this. Okay. Between the phloem and the source cell, there are companion cell. Okay. The companion cell will pump the hydrogen ion to the source cell through the proton pump. So you need energy. So it's active transport. Actually, the active transport happening for the pumping of not the ion, hydrogen ion. Okay. And the hydrogen ion gradient is set up between the companion cell and the source cell. More hydrogen ion there. Hydrogen ion moves through the membrane down about the concentration gradient. And then by the co-transport, the organic compounds that move to the phloem through the companion cell with hydrogen ion. A high concentration of the organic compound is developed in the phloem beside the source. So it's very generous. We move the light with the hydrogen ion to the right. And the light hydrogen ion will move the heavy ion with it through co-transport. Very smart design. Okay. Adaptation of fireman sieve tube. Okay. Sieve plate. Okay. Block the contamination larger than the small organic compound in the sap. Okay, reduces cytoplasm organelle and no leakage loving daily. Okay, for the sieve tube. Okay, element to increase the size of the channel for the flow of cell, but the phloem is still a living cell, not the Xylem is a dead cell. Okay, Presence of many mitochondria for compelling cell and plasmodesmata between them to provide energy for active transport. Comparison between Xylem and Phloem. You know it. Okay? Xylem has lignin. Okay? So its vertical cross section also has horizontal lignin. Phloem does not have lignin. Okay? It doesn't have so much high pressure to be support. Okay? Its vertical cross section is clearly hollow. Xylem transport water from roots to leaf. Phloem transport sugars from leaf to roots. Okay, opposite way. Opposite direction, different way. Xylem process is called transpiration. Phloem is called translocation. Xylem only has water. Phloem has a high sugar concentration. Xylem uses osmosis. Phloem uses active transport to draw sugar from the leaf, okay, and then hydrostatic pressure down the plant. Okay. Xylem vessel is wider and with a thicker wall than phloem sieve tube. Okay. Xylem is a dead cell. Phloem is still living cell. Only phloem has a plasma membrane, sieve tube, and companion cell. Xylem has nothing, just hollow thing. How to measure the phloem transport rate? APS stylus, fairly smart way. Okay. APS is a small insect sucking a sap from phloem to get the nutrient. Okay. APS uses a stylet to tap into the phloem. After inserting the stylet into the phloem, APS cut it out from its body, so the phloem sap continues to flow out from the stylet, and then the APS can drink from it. Okay. Very smart way. So how can we estimate the flow of sugars? Okay. How can you see the sugars? Okay, because sugars has carbon, right? So we use a radioactive carbon. Okay, carbon 14 is a radioactive carbon. Okay, we have plants here, and then we have a radioactive carbon dioxide here, and then the plants absorb this radioactive carbon dioxide, becomes radioactive sugars. When the sugars flows through the plants, we can detect the radioactivity to know where or how far did the sugars go. Okay. And all the thing we like is, okay, we put a cross section of the xylem and put a sealed X-ray film on it. And after some time, we will find out that, oh, the black dot are there. The black dot is the phloem phase. So we conclude that the sugar is flowing through the phloem, not the xylem. And thank you for going through with me. So today we completed, okay? We complete the understanding of transport of blood, the heart system, and also the transport of sugars, organic compound, from the source to the sink in the plant, okay, the translocation. So thank you for going through me. I hope it helped you a little bit, okay. If you like our share, remember like, subscribe, and share with friends. Okay. See you. Bye.